let's go back to equilibrium in hearing. So the inner ear obviously takes care of hearing, but also allows us to remain sitting in a chair or sitting in a car or walking in dynamic equilibrium. We're constantly aware of what position of our head is and the position of our body. Uh, so that's the uh, functions of the ear. We already did the visual pathway. Okay, now we're going to, the ear has chambers filled with endolymph and chambers filled with perilymph. These fluids are very important for the supplying the ions and the opening and closing of channels when sound generates action potentials in your inner ear. So let's look at a picture of the ear. So you have the auricle, the outer ear, the external acoustic canal, the eardrum or the tympanic membrane, the auditory ossicles in the middle ear, the cochlea, which is the organ of hearing, and then the semicircular canals, along with the vestibule and the parts below the semicircular canals. The auditory ossicles take sound waves and magnify them and vibrate on the oval window. That vibration is sent into the cochlea. It's called that because it looks like a shell, a seashell. And the oval window is where the energy of the sound wave is allowed to be dissipated so that the sound wave is now finished and you can have additional sound coming into the cochlea. So the vestibular cochlear nerve has a vestibular branch and a cochlear branch. The cochlear branch is involved in the process of hearing. The vestibular branch is involved in the processes of equilibrium. So the receptors for hearing are in the cochlea, the semicircular canals, the vestibule, and other structures below the semicircular canals are involved in balance and static balance and dynamic balance. So here's the eardrum. Here are the malleus, incus, and stapes. Their job is to amplify the sound waves and to transmit that motion into the cochlea. The round window is, as I said, for dampening or making sure that the sound waves don't persist and reflect back through the cochlea. They have to undergo their pathway in one direction and then be dissipated at this round window. Otherwise, we wouldn't have separation of sound waves and we couldn't hear. The auditory tube is called the eustachian tube that connects the middle ear to your not to your cavities in your pharynx and in your pharynx. So you know you can equalize pressure when you have changes in pressure in this tympanic membrane. So if we look more at the cochlea and we look more at the semicircular canals and the vestibule and the utricle and the saccule. So these semicircular canals and these vestibules, utricle, and saccules are going to have hairs that change in response to little rocks that are going to cause changes in the impulses coming from these equilibrium centers, and it's going to send that information back to the brain, obviously, but it's going to generate action potentials to tell you the position of your head and the dynamic movement as well as static movement. So the hearing part of the inner ear is the cochlea. The organ of Corti is the organ where the auditory receptors are. And we'll find out that these semicircular canals separate endolymph from perilymph, and their movement is converted into nerve signals by small little rocks generating action potentials to tell you about the position of your ear, your head in both static and dynamic environments. So within the anatomy of the ear, you have hair cells, and displacement of these stereocilia, depending upon how sound waves travel in the cochlea, you can either stimulate the hair cell or turn it off. So the movement of these stereocilia is going to lead to nerve impulses sending information about hearing to the auditory center. The equilibrium is taken care of by the semicircular ducts, and you have various structures which we'll point out, but the gelatinous mass is distorted by fluid, and that's going to help us get information in three planes: anterior, posterior, and lateral. And that's going to be in the saccule, as I said, in the utricle. And these hair cells are going to be stimulated by otoliths, heavy mineral crystals in a thick gel. And gravity will change the position of these otoliths, and that's going to lead to nerve impulses leaving the equilibrium center of the semicircular canals in the saccule and the utricle, and that's going to have these hair cells. And in the vestibular complex, the vestibule, you can have movement of this cupula based upon the otoliths, and that's going to change the position of these hair cells, and that's in the semicircular duct. And the direction of rotation can be recorded, and we have a sensation of head movement and rotation. So you have these little otoliths that are in the vestibule, and these otoliths are what's going to change the position of these hairs based upon the bending of the cupula. So that's the vestibular complex. Back here, the vestibular duct and the part of the structures below the saccule and the utricle. So depending on your position of your head and the otoliths, that's going to cause nerve impulses in these nerve cells, in these special receptors, and it's based upon the gravity effect on these otoliths, and the movement of these hair cells sends information down your eighth vestibular nerve, and that's going to be analyzed in your brain, and you're aware of the position of your head. Okay, so how does hearing take place? So what's going to happen is that you're going to have the cochlea, a cross section of the cochlea, and you're going to have these various vestibular ducts, the organ of Corti, and the tympanic duct, and there'll be questions on these structures on WebAssign, and the cochlear duct. So the vibrations that enter the cochlea from the auditory ossicles, and which enters the cochlea, is going to cause distortion on various parts of this organ of Corti. And that's going to be the organ of Corti, and it's going to have hair cells, and above it is the vestibular membrane and the tympanic duct and so on, the cochlear duct. And the movement caused by sound waves, which has been amplified by the auditory ossicles, is going to cause bending of these these hair cells in the basilar membrane. So the basilar membrane is going to be vibrating at a particular location, and depending upon the location where the sound waves cause vibration in these hair cells, that's where you're going to be able to pick up the loudness and the pitch of the sound. So the frequency and the loudness. Okay, so here we have a step-by-step entry of sound into the external canal, the amplification of the sound through the auditory ossicles into the oval window, and then those waves in the fluids of the cochlea are going to cause vibrations at particular locations along the basilar membrane. And remember that round window is going to allow the dissipation or the dampening of the wave so that you don't have reverberations and back echoing, which is going to make it very difficult to be able to determine individual pitches and loudness of the components of sound that your middle ear and outer ear are exposed to. So movement of the tympanic membrane is going to cause movement of the auditory ossicles. The movement of this stapes on the oval window establishes a pressure wave in the vestibular duct. The pressure waves distort the basilar membrane, which has the organ of Corti all along it. The vibrations cause hair cells to vibration against the tectorial membrane, and information about the intensity, the loudness, and the frequency is conveyed to the central nervous system based upon the location of these waves on the organ of Corti on the basilar membrane. So here we have this basilar membrane, and we have the receptor cells, and here's the tectorial membrane that is on top of all of these cells, and this bending and the interaction with sound and the tectorial membrane is going to cause movement in the basilar membrane, and depending where on the whole length of the cochlea, so the whole length of the cochlea, different messages of pitch and loudness, or frequency and amplitude, are going to cause movement in this basilar membrane, and that's going to be picked up by the receptor cells, and that's how you hear. So the auditory pathways start with hair cells, sensory neurons to the spiral ganglion, and afferent axons through the cochlear branch, synapses in the medulla, and relay through the midbrain and thalamus into the auditory cortex in the temporal lobe. So let's stop there, that recording. Stop recording, stop recording.